Okay, so here we're going to look at the mitochondria and we're going to focus on what happens during the citric acid cycle or the Krebs cycle. So the mitochondria, because it evolved from ancestral prokaryotes, has a double membrane structure. It has an outer membrane, that's a lipid bilayer, as well as an inner membrane. And you can see here, I've color-coded it, where you have the space between the two membranes in like a blue color, and then this area is the matrix, this orangish yellow color. Now it's within this orangish yellow color, this matrix area, that the Krebs cycle or the citric acid cycle is going to occur. So we have the intermembrane space, that is basically where the proton gradient becomes established during the electron transport chain, and then the Krebs cycle is what happens in the matrix. So let's pick up where glycolysis ended. So at the end of glycolysis, the products are 2 pyruvic acids or pyruvates, 2 ATPs, and 2 reduced electron carriers, NADPH. I'm sorry, NADH. So now, as we go into the mitochondria, when it is aerobic respiration, one of the first things that's going to happen is we're going to oxidize this pyruvate. Now this step, the oxidation of pyruvate, is sometimes attached to the Krebs cycle or citric acid cycle in textbooks. So it'll be taught as one process. So if we remember that the whole process of aerobic respiration is to fully oxidize our food, if you remember, oxidize means to remove electrons. And so what's going to happen here is we're going to oxidize the pyruvate, which means we're going to break the bonds and we're going to take the electrons. So oxidize pyruvate, and at the same time we're going to reduce that electron carrier. That electron carrier gained electrons and a hydrogen, and we'll carry that over to the electron transport chain. Now in the process of breaking that bond, a carbon dioxide was released. This carbon dioxide is the carbon dioxide that you breathe out. Now we're left with 2 carbons remaining in an acetyl group. So then a coenzyme will come and attach to that acetyl group, or acetyl, and now that together, acetyl-CoA, is what will enter into the Krebs cycle. So the oxidation of pyruvate happened within the mitochondria, and now we're going to continue and zoom in to see, well, what's exactly happening to that acetyl-CoA. Since there's still bonds that can be broken, there's still potential energy between those carbons. So when I look here, I have 2 reduced electron carriers from glycolysis, and then I have 2 that were just made in the oxidation of pyruvate. I know we only saw 1, but really there's 2 pyruvates made at the end of glycolysis, so there's 2 reduced electron carriers. So in the process of the Krebs cycle or the citric acid cycle, what we're going to do is we're going to need to get our food, the remaining carbons, fully oxidized, right? So what we're going to do is we're going to first have an enzyme help attach this acetyl to a 4-carbon molecule, and then the coenzyme can be used again. Now here we get a 6-carbon molecule called citric acid. And so the first step of the Krebs cycle, an enzyme will attach the acetyl group to a 4-carbon molecule forming citric acid. And that's why this is also called the citric acid cycle. So here, our next step, now in reality, the Krebs cycle actually has 8 enzyme-controlled reactions, and we're going to just gloss over the main parts of the Krebs cycle, the overall big picture. So we have potential energy still available in those bonds, so we're going to oxidize this citric acid, breaking off a carbon dioxide, and then reducing the electron carrier. Then we're going to do another reaction where this substrate will be oxidized by an enzyme. There goes our carbon dioxide, another reduced electron carrier. And then in a series of reactions, you get 1 ATP made by substrate level phosphorylation. And then in the next enzyme-controlled reaction, we get a new electron carrier called FAD2+, that will actually be reduced into FADH2. And then in another reaction controlled by enzymes, we get another reduced electron carrier. So this process, throughout the Krebs cycle, we're now back with those 4 carbons that we started with. So as a cycle, it brings us back to the start. Oh, so here you can see it's actually a very complicated process that includes a series of enzyme-controlled reactions to oxidize our food into carbon dioxide and get the potential energy into the form of electrons. So our next step, we could actually start the Krebs cycle all over again with the next acetyl from that second pyruvate. So once we go through the Krebs cycle a second time, by the end of this, we will have a whole lot of reduced electron carriers. So the purpose of the Krebs cycle, if we really think about the big picture, is to break those bonds in our food, releasing that potential energy. Some of it will be converted off and given off as heat, and then the energy in the form of electrons will be carried over to the electron transport chain. So by the end of the Krebs cycle, our glucose, our food, is now completely in the form of carbon dioxide, and the electrons are stored in electron carriers. So the Krebs cycle's purpose is to fully oxidize your food and have a whole bunch of reduced electron carriers. Now, this is pretty rad because if we look at what's happening here, you had your glycolysis that made pyruvate. That pyruvate became acetyl-CoA in the matrix and then went through the Krebs cycle or citric acid cycle, making 1 ATP. But now the next step is the electron transport chain. And that electron transport chain is located in this inner membrane, or these cristae. So now you have a whole bunch of reduced electron carriers that are right there and readily available for the electron transport chain to establish that proton gradient in this orange space that will then lead to chemiosmosis and oxidative phosphorylation.